Good morning and welcome back to this channel, StudentSOS. Or if you're new here, very good welcome to you all. Now this morning, I'm already aware that I'm going to be in trouble for not having my face on this video. However, I'm going to tell you that that's a bonus for me, and trust me when I tell you that it'll be a bonus for you too, viewers. And anyway, looking at my face doesn't help you learn biology. I'm hopeful this will though. I will be making no apology for beginning this video series with Year 12 revision. And we're going to take a look at some of the topics you will have looked at right at the start of Year 12, which you've probably long since forgotten about. So here we go. We're going to take a look this morning at how we get from DNA to a protein. This will incorporate many of the organelles in animal cells that you need to know about. Please make sure that you know exactly what these structures do and which of them are found in animal cells or plant cells or both, and also which are found in prokaryotes and eukaryotes. More on that to come shortly, but let's dive in. And we're going to begin in the nucleus with the DNA. What I'm going to say here is that there's a 1 inside that nucleus, and that's for a reason. I have a set of notes on this that I'm going to make available, and if you like these, please place a comment below, because I'm actually planning on making available a full set for AS and A2 for topics, including all of the notes. So I'm going to make those available as sets if there's interest for them. So inside the nucleus we have the DNA. Here it is, double helix. And we use the DNA, of course, because it has genes in it which code for proteins. And that's very important that we know that right at the beginning. So we're going to code for proteins here. You'll notice that there's a nuclear membrane around the nucleus. This is made of the exact same material as the plasma membrane or cell surface membrane. Please note, Year 12, we do not call it a cell membrane at A level. You will not get the mark. So this is made of the exact same material, but we also have, as I've labelled here in the diagram, a nuclear pore. And in fact, there are a number of these pores, and you can already see that my little piece of mRNA is sneaking its way out through a nuclear pore. But let's begin at the beginning. So the DNA is instructed to unwind and unzip at a certain section, a gene. We'll talk more about why and how that happens in Year 13. So then we allow RNA polymerase, this is an enzyme, to attach to a start codon or a start region of the gene, a promoter sequence. And then we add free nucleotides, which have come from the nucleolus, of mRNA. Please note here that mRNA and DNA have slightly different bases. So in DNA, the bases are A, and that pairs with T, and G, which pairs with C. In mRNA, and in fact in all RNAs, you need to remember there are no T's. In their place is the letter U for uracil. So when we have an A in the DNA, we place a U opposite it in place of the T. This is complementary base pairing. So once we have used RNA polymerase to complementary base pair with the sense strand of DNA. If you're not sure what that is, skip forward to look at transcription. The sense strand of DNA, and we make a copy, an mRNA copy of the gene. And indeed, there we go, little single-stranded mRNA copy of the gene. There it is, and out it goes out of the nuclear pore. And from here, we move to a ribosome. And at the ribosome, we've got the mRNA, which goes in between two halves of the ribosome. Here you can clearly see a number 2, and the number 2 is again linked to the notes on this topic. So everything that I'm saying will be available down below in the notes section. So the ribosome then reads this piece of mRNA, and it reads it in sets of 3. We have sets of 3 called triplets or codons, and they read 3 letters at a time. And then translation is the process by which tRNA molecules have an anticodon that links to the mRNA, and that attaches an amino acid to a growing chain at the top of the ribosome. And what you should be able to see there is the product. There's my protein product. And that protein will do some folding. I am not sure why year 12, but many of you seem to have huge nightmares with protein structure. Please look at this carefully. We'll do a separate video on this because it seems to be a regularly struggling topic. I think it's probably because it's not very exciting. We'll excite it up for you. We'll get it exciting for you. So that protein is now going to move to the Golgi body or the Golgi apparatus. There it is. Doesn't matter which you call it. And here we're going to add something, for example, like a carbohydrate group, or indeed a lipid group, or both. And you can see here on this diagram, this particular protein has had a couple of blue lipid groups and a little yellow carbohydrate group added to it. The Golgi body is responsible for what we refer to as post-translational modification. Now that sounds very scary, but actually what it means is once the ribosome has done with translation, we can modify my protein as such so that it can do whatever it is that it's destined to do. So the protein may or may not require modification, but the Golgi body is responsible for packaging into vesicles. You can see this little protein here is inside a vesicle. And this vesicle then is able then to do one of a number of things. It can move to the cell surface membrane, just here, and that would be for the purposes of exocytosis or secretion. A good example of this would be the insulin hormone, but there are lots of other examples, including pretty much all the hormones. But it could also be used inside of the cell, and I haven't drawn them on here because that would make the whole diagram look quite messy. But what we have is a surface covered in cytoskeleton. This is like an enormous scaffold that runs all the way through the cell, and you'll have heard of cytoskeleton, and it's probably something you've completely ignored. Please don't ignore it. It's very important. It allows things like vesicles to move to the cell surface membrane, organelles to move around the cell, and all kinds of other jobs. So if you've looked at mitosis this year, it'll allow chromosomes to separate. That's what spindle fibres are made out of, cytoskeleton. So that's a really important component, but the fact that you don't learn lots about it means that you probably remember absolutely nothing about cytoskeleton. Please ensure you have an understanding of that. Okay, we also have on the diagram a mitochondria. Now, in year 7, you'll have called this the powerhouse of the cell, and unfortunately you may have done that at GCSE as well. It is a powerhouse, but please don't use that language at A level. The mitochondria are responsible for aerobic respiration, specifically aerobic respiration. You'll learn much more about this either in your course now, after your current exam series, or indeed during year 13. But that's what the mitochondria do. They provide the energy for you to be able to do all the things that we've discussed so far in this video and many more. And we also have some smooth endoplasmic reticulum. This is another component of the cell that very few students are able to answer in an exam what it does. It is responsible for things like lipid synthesis and transportation. So steroid hormones, these kinds of things. This is the role of the smooth endoplasmic reticulum. The rough endoplasmic reticulum is usually found very close to the nucleus membrane, and indeed if you have been looking at electron micrographs, more on that soon, then you will find that there's a lot of rough endoplasmic reticulum that surrounds the nuclear membrane. If we zoom in a little bit here, you'll see that there are little red dots on here, and these are of course your ribosomes, and I kept the colour the same as my ribosome here. These are just much smaller versions of them on the diagram. So the ribosomes that we use for translation and therefore production of protein can either be free in the cytoplasm or indeed attached to endoplasmic reticulum. The advantage of the endoplasmic reticulum is that it will also allow transport around the cell of the completed protein. But that completes this particular section of work on a run around the cell and some of the major organelles and how we get from DNA to a protein. Please like and subscribe if you enjoyed this video. Drop a comment below of anything else that you'd like to ask about it, or indeed if there are any specific details of any organelles you'd like to know about. Thanks very much for watching. Have a great day.